Hi, my name is Shireen Yates. I'm the founder and CEO of Six Sensor Labs, a company that's dedicated to helping people live happier and healthier lives through food transparency. To our team, food transparency means having better data about what's in your food so you can make more informed decisions about properly nourishing yourself. I was diagnosed with many food allergies and food sensitivities about 10 years ago and found it extremely challenging to eat socially and stay healthy. Leading up to my diagnosis, I was very sick for many years. I felt consistently subpar, and even when I was properly diagnosed with a number of different food allergies and sensitivities, I found it incredibly challenging and nearly impossible to eat out and stay healthy by avoiding those foods. Observing the growing population of people with food allergies, I was inspired to dedicate my life to providing tools to help people with food restrictions. I founded Six Sensor Labs, and our first product is a portable gluten sensor designed for consumers to use on the go. Today, it is my absolute pleasure to introduce Dr. Jingqing Zhang. I had the honor of meeting Dr. Zhang in graduate school at MIT, where we shared a passion for helping people with food restrictions. She joined the Six Sensor Labs team in 2013, and I'll now hand it off to her so she can tell you more about her background.
Hi, everyone. My name is Jing Qing. So I did my PhD in the Department of Chemical Engineering at MIT and studied nanotechnology. After graduation, I joined a glucose diagnostic company called AGA Matrix, creating glucose test strips and sensors for diabetic patients. When an opportunity arose, I joined Six Sensor Labs early last year. So why did I join Six Sensor Labs? I was initially drawn to Six Sensor Labs out of a personal need, since my husband actually has celiac disease, and we frequently found it very difficult to navigate the gluten-free diet, both at home and when we were traveling or eating out socially. I met Shireen while we were both at MIT, and after speaking with her and many other people with celiac or gluten intolerance in my circle of friends, I quickly realized that this was a problem that was shared with many others. And this really motivated me to join Six Sensor Labs full-time to develop a test that can really help millions of others. So how does NEMA really work? The beauty of the device is its simplicity. In 3 easy steps and in 2 minutes, the NEMA device can test a sample for gluten and provide the user with an easy-to-interpret result. The NEMA is comprised of 2 parts, a disposable cartridge and a sensor. The first is a disposable test cartridge, which contains the chemistry required to detect gluten and the other allergens that we will be developing in the future. The second is a reusable sensor, which aids in performing the assay, evaluates the test result, and provides user with a binary yes or no result. With these 2 components, the users can perform laboratory-grade testing in your hands. In order to operate the NEMA device, the user simply places a food sample into the test cartridge, screws on the cartridge cap, puts the cartridge into the sensor device. In 2 minutes or less, if the NEMA detects any gluten, it will display a visual signal to let you know the presence of gluten. If NEMA does not detect gluten, the sample you just tested is considered to contain at most 20 parts per million or 20 ppm of gluten, which is defined as gluten-free by the FDA. So what foods can we really test? As someone who has to navigate a gluten-free diet with my husband, I realized that it is very important for the NEMA device to be able to test as wide of a range of foods as possible. And this is something that our research department is constantly working to improve. So far, we've demonstrated successful testing with a range of solid and liquid foods. Specifically for homemade foods, some of the solids that we've tested include meat and veggie burgers, oatmeal, cereal, breads, cupcakes, chips, and muffins. And some of the liquids include thick and thin matrices, such as sauces and soups. And for restaurant foods, we've successfully tested such items as rice, meat, bread, sandwich, noodles, cooked rice, beans, salads, donuts, dips, tacos, omelets, and cookies. We posted a few restaurant testing blog posts in which you can read about our findings from these studies in greater detail, and we will continue to test more as we further develop our product, and we will continue to publish our results. While we've had many testing successes, we do know that our device does have limitations, and it has difficulty detecting gluten in fermented drinks such as wine or beer. So this is something that we are continuously working to improve. When we finally launch our device, it will come with a list of food that we have fully validated in our lab. So how does the chemistry work? The detection is based on a type of assay called immunoassay. These assays take advantage of a unique class of proteins called antibodies, which are able to specifically recognize a single target of interest, in our case, gluten. A small amount of gluten will trigger the antibody to bind to the gluten molecule, thus causing an optical response or a color change in our assay, which is then further captured by an electronic reader that is built in the device. In order to achieve the sensitivity and specificity that we want in the device, we spent a year developing our own custom antibody against gluten. We're aiming to detect gluten at 20 parts per million, meaning if the food has a gluten level of exactly 20 parts per million or 20 ppm, our device is designed to be able to detect at 99.5% of the time. The exact accuracy will continuously to be evaluated during the next stage of testing, and we aim to publish the data before we ship the product. If the food contains gluten at a level higher than 20 ppm, we will be able to detect it with an even higher accuracy than 99.5%. If the device detects gluten at any level, it will display it on the screen. Specifically regarding cross-contamination, NEMA takes a sample from your dish. The risk of a cross-contamination remains for any food you did not sample. However, NEMA adds another layer of data about the food when making your dining decisions. So why do we decide that we want to work with 20 ppm? On August 5th, 2013, the FDA issued a gluten-free labeling rule specifying that a food can only be labeled as gluten-free if it contains less than 20 parts per million or 20 ppm of gluten. While different individuals almost certainly have different sensitivities to gluten, there is currently no way to determine a gluten tolerance level on an individual basis. So this FDA ruling is the best guideline that we currently have for evaluating which foods are safe for gluten-sensitive or celiac individuals. We are currently following ISO certification during our development and manufacturing processes. We are also largely following AOAC guidelines on food allergens and gluten testing. Generally, there are a series of standards that help us to ensure that our products are of the highest quality. We have a roadmap for NEMA. We are creating food transparency at Six Sensor Labs. We believe that food can have an enormous impact on your daily life and well-being, and we want to ensure users with the ability to be their healthiest self by knowing what they eat. As a starting point, we're aiming to provide a tool to people who are avoiding certain foods due to allergies or food intolerance, and we hope to provide these individuals with a product that can be seamlessly integrated into their daily life. We're aiming to detect all 8 allergens defined by the FDA, which include wheat, milk, eggs, fish, shellfish, tree nuts, peanuts, and soybeans in the near future. Gluten is technically not an allergen, but it is our first target. Our first 2 other targets after gluten would be milk and peanuts, and we are already working in the lab on these 2 allergens. One challenge is that regulatory agencies have not yet defined any thresholds regarding what allergen levels are sufficient to cause an adverse response in the allergic population. Without a better understanding of such a threshold, it is very difficult for us to figure out what limit detection we are targeting in our assay. Therefore, we are consulting with regulatory agencies to better understand the problem and to propose a reasonable limit detection that will cover a majority of the cases. For example, if 99% of the peanut allergic population is responding to a peanut level at 0.1 microgram per mil, we should try to meet or exceed this level of detection in our test. Our first goal is to develop a robust and reliable assay for each individual allergen. After that, we hope to utilize what we've learned to develop a multiplex technology. Each allergen requires a different chemical composition of solution to extract the allergic protein. If we want to multiplex, we will have to figure out a universal extraction solution that would extract all allergens at once, which is very challenging to do. The second challenge we're facing is that the detection of each allergen requires its own real estate in the device. And because we want to keep the device very small and portable, we have limited space for multiplexing. However, this is something that is definitely in the pipeline as we continue to develop our product. And we are here to open for questions.
Well, thank you so much, Dr. Zhang, and we also wanted to thank everyone who attended our webinar today. If you have any follow-up questions, please feel free to reach out to us on Twitter. We're at MimaSensor or at 6Sensor Labs, and you can also send us an email, mimasupport@6sensorlabs.com. Thank you so much, and hope to talk to you soon.